Welcome. In this session, I'm going to discuss about a human genome project. So in short, it is called HGP. Now let's see what is genome. So genome is haploid set of chromosomes in a cell. So all that haploid set of chromosomes in a cell will be called genome. Now, this human genome project, it was a mega project. So why it is called mega project? Okay. So why it's called mega project? So we know that the human genome has 3 into 10 to the power of 9 base pairs, okay, are present in human genome. So these are the number of base pairs, okay, present. Of course, you can take 3 into, 3.3 into 10 to the power of 9 base pairs are actually present in human genome, right? So here I just taken 3 into 10 to the power of 9 base pairs in human genome. And to sequence, right, so a base pair, right? So to sequence one base pair, it would require 3 US dollars. Okay, so per base pair, 3 US dollars are required. So the amount of money that is required for finding out, okay, one base pair will be 3 US dollars, right? And with this calculating, so 3, multiplying with this number of base pairs, it will be around 9 US billion dollars. So that will be, right, 9, okay, billion dollars are required, okay, for this project, right? So for that reason, the Human Genome Project is called mega project, right? So the number of base pairs present in the human genome, so 3 into 10 to the power of 9, and for finding out one base pair, the amount of money required is 3 US dollars. And okay, the total amount of money to complete this genome, right, will require 9 billion US dollars. So for that reason, the project is called mega project, right? Now, once we find out the sequences, so what happens, how the information will be once we sequence the DNA. So it means that we know if it is the DNA, we need to find out the sequence right from one point, okay, say AG. So this is the sequence. So we need to know one strand because the other strand is complementary. So no need to write the information of both the strands. So one strand information is more than enough because as the two strands are complementary to each other. And now I'm going to isolate or find out these sequences and then need to put it, okay, in a book, say suppose AGCCTACC. So I will be going on in this way. Okay, so you can see that whatever that sequence is, I need to put it on a paper and say suppose I'm going to write this information in book, then I would require 3,000 books. So, the total number of books that I would be requiring will be, of course, more than 3,000 books. So I would be requiring 3,300 books, and each book should have, and each book should have 1,000 pages. So each book should have 1,000 pages. So now later, even though if we store all that information in the books, okay, each book having 1,000 pages, so later to find out the information from those books will be very quite difficult. So hence scientists, what they have done is whatever that information, okay, after sequencing the human DNA, so they have kept that information into a computer. Okay, so all the information was loaded into a computer. So here, at this point, what happened is the biology came up with the computer, and that resulted in evolution of a new field in biology called bioinformatics. So the biology plus computers resulted in bioinformatics. So it is a new branch of biology that deals with, okay, biology and computers. So whatever that information we obtained, okay, while sequencing, and of course later we need to find out, okay, what is the sequence of a particular gene, okay, and where is the position of that gene, whether it is present on the chromosome 1 or chromosome 2 or on chromosome X or chromosome Y. So that what we call it uh, maps, right? So here we need to find out the locations or the positions of genes and sequences of genes. All that information, if we store it in the book, it's very quite difficult to get that information back. Okay, so for that reason, we have kept that information in computers, and we developed some special programs that actually help in storing that information and then retrieving the information, right? And this project, so that was started in 1990. Actually, this project was a 13-year project. So it took 13 years, okay, to complete this project, right? And the funds for this project came from different sources. So the funds, the money for this project, so from where the funds came to this project, so the funds came from different sources, as I said. Okay, then here actually the project was funded by US Department of Energy. So US Department of Energy, right? And also included with is National Institute of Health. Okay, so it's called NIH, National Institute of Health, and Department, US Department of Energy. So they have, okay, funded starting in the beginning of this project. These two have funded, okay, organizations, they have funded this mega project, and then later, right? Oh, Welcome Trust. So and another company, okay, Welcome Trust. So this Welcome Trust from UK. So this became major partner for this. So Welcome Trust from UK became major partner in this case. And then of course additional contributions came from different countries. So additional contributions came from Germany, from Japan, right, and from, okay, China, France, etc. So from different countries also, money, that is fund, has come to this project, and this project was completed. So project was completed in 2003. So the project was completed in 2003. Right, but the chromosome, the last chromosome to be sequenced, last chromosome to be, okay, or last chromosome sequenced was chromosome number 1. So that is the biggest chromosome. Later I will come to that point. So last chromosome sequenced was chromosome number 1, and that was finished in, so that was finished in May 2006. Okay, right, out of the 24 chromosomes in human beings. Okay, so that 24 chromosomes in human beings will be 22 autosomes plus X chromosome plus Y chromosome. So that will make it total of 24 chromosomes. Okay, so 22, 23, and 24 chromosomes. Out of this, the last chromosome sequenced was chromosome number 1, and that sequencing finished in May 2006. And once we finished with human beings, we also started sequencing the DNA of other organisms also. So once we finished about sequencing the human DNA, right, we also sequenced DNA of other organisms. So DNA of other organisms, in which it included, okay, uh, bacteria. So that is the E. coli bacteria, and then yeast. Okay, so Saccharomyces cerevisiae. So okay, so its DNA is sequenced, and then an, okay, a non-pathogenic nematode. So it's called Cenorhabditis elegans. So Cenorhabditis elegans. So this is a non-pathogenic nematode, and it is free living. It's a free living, non-pathogenic means it does not cause disease. So free living non-pathogenic nematode. Okay, so the that organism's DNA also we have sequenced, right? And then we also sequenced the Drosophila, the fruit fly DNA is also sequenced. And then coming to plants, in case of plants, we have sequenced the rice, the Oryza sativa, so whose DNA is also sequenced, and then Arabidopsis is another plant. Okay, so these two are plants. So the remaining all, okay, so of course this is bacteria, this is fungi, and then this is a nematode, okay, this is an insect, and these are plants. So all for all these organisms we have sequenced DNA, right? Now let's see how they have sequenced the DNA of human being or other organisms. That's what we see about uh, methodologies. Okay, of course, before I discuss about that methodologies, I would like to first discuss about objectives or goals of HGP. So objectives or goals of HGP, right? So here the objective of HGP is to identify all genes in human beings. So to identify all genes in human beings, right? Of course, identifying all those human genes can help in treatments of different diseases, and the way the doctors now treating can change in future, right? And then even to determine the 3 billion nucleotide sequences, okay, to determine 3 billion nucleotide sequence, right? And then the next one, to know the location of genes, to know the location of genes, right? So that's a mapping. So chromosomal mapping, what we call the chromosomal mapping, will indicate the positions of genes on the chromosomes. So when you see a chromosome, so when you see a chromosome in this way, say suppose from this point, okay, we have some gene. So and from here this is another gene. Okay, but don't think that all the sequences on the DNA will be coding or will be a gene. So there will be some genes that doesn't code for anything, right? That later we will see. So here is a gene and here is another one. So like this, the different genes, we call it chromosomal mapping. So to see the locations of genes on chromosome, okay, so locations of genes on chromosome. So that's to know the location of genes on chromosome, and then to acquire complete knowledge of organization, to acquire complete knowledge of organization. So how it is organized, okay, so everything in detail we want to know, and the structure and function. So the structure and function of human chromosomes, right? So that is the one. And store this information in computers, store this information in computers. So that is what, okay, of course, that we call the database. So the database, whatever that information we store, okay, so that will be stored in the databases of computers, okay. And then even to identify ethical, so to identify ethical, legal, then social implications. So to identify the legal, so any legal or any ethical or social implications that may rise from a project, that may rise from the project, okay. So these are some of the objectives or goals of human genome project, right? So now we will see the different methods that are used in sequencing the DNA. So let's see the methods used to sequence DNA. So here they follow two methods. One is expressed sequence tags or expressed sequence tags. So in short, you can call so ESTs, expressed sequence tags, and then the second one is sequence annotation. So these are the two methods they have followed to sequence the complete DNA. So here, what is the expressed sequence tags? So let's see, we know that uh, in case of DNA, not all portions of the DNA will be active all the time. So there are, and moreover, there will be non-coding regions also. So say suppose from this point to this point we have some gene, and this gene will get, say suppose, transcribed. So it is transcribed and results in the formation of mRNA, right? And say suppose here to here, of course the length of the gene varies. Not all the genes will be having same length. Even though if they have same length, they may not be having same sequences, right? So here this is another gene, and again transcribed and may produce mRNA. Now what we did in expressed sequence tags, we have sequenced these mRNA molecules. Okay, so here uh, the expressed sequence tag is a short subsequence of cDNA, that is complementary DNA. From this mRNA, we are going to make the DNA molecule, and that's called cDNA. So here the C stands for complementary DNA. Of course, by knowing the sequences on mRNA, we can predict the sequences on the DNA also. So by making a DNA, so whatever that DNA we make from mRNAs, so we usually call cDNAs, complementary DNA sequences. Okay, and these complementary DNA will be used for determining the gene sequences. Definitely, this mRNA, when it is transcribed, we know it is going to get translated and produce a polypeptide. So that's what we say gene expressed, and it means that if you are sequencing mRNA, actually you are knowing the gene sequence, right? So first, like this they have done, okay, in expressed sequence a tags method. So they have sequenced all these mRNAs, right? And this one helped in determining, okay, so it helps in determining gene sequence. So that one helped in determining, okay, the gene sequences, right? So that's what we see in case of expressed sequence tags. Now coming to sequence annotation. So what we have followed in sequence annotation. Okay, so in case of sequence annotation, what they have done, starting from the starting point of the DNA to the end point of the DNA, they continuously started finding out the sequences. So in this case, that is express tags, you will not be knowing the sequences that are present between the genes. Okay, so if this is not expressed or it has some non-coding sequence, so this non-coding sequence will not be known in case of this expressed sequence tags. But in case of the next method called sequence annotation, so it will be sequencing the DNA from the start point to end point. Okay, and this will include coding and non-coding sequences. So it includes coding and non-coding sequences. Okay, right? So this is how we see from the uh, start point to end point continuously, okay, including all the coding and non-coding sequences, but in this case only the coding sequences. Okay, so that's what only the coding sequences we identified, but in sequence annotation it included both coding plus non-coding. Now, one problem in sequence annotation is that you cannot sequence the whole DNA at once. So you need to, okay, break this DNA into fragments, and those fragments need to be cloned. And in this case, we require something called hosts and vectors. So in case of sequence annotation, so we require hosts and vectors. Okay, so here I'm going to break down the DNA molecule, okay, into fragments. So break down the DNA into fragments, and each of these fragment is need to be, okay, joined with a vector. Okay, so and then the vector, here in the vectors that are used are usually BAC and YAC. So this BAC stands for bacterial artificial chromosomes, and YAC stands for yeast artificial chromosomes. So bacterial artificial chromosomes and yeast artificial chromosomes. So once this fragment of DNA is linked with this vector, so the vectors, okay, will be helping in transferring this fragment into the host. And the host we used here are bacteria and yeast. So bacteria and yeast. So this BSC will help in transferring this one into the host bacteria, and YSC will help in, okay, to deliver it into yeast. Okay, so these vectors can help in the process of cloning. That's what, okay, so here linking of these DNAs, that is the fragments of DNA to the vectors. Okay, so it's what we call cloning, and then putting them into the bacteria, right? And this actually helps in determining the sequences. Of course, later we will be finding out these DNA sequences by using automated DNA sequencers. So there are some machines, and those automated DNA sequencers will be following a principle that was developed by Frederick Sanger. So later we use automated DNA sequencers. So these automated DNA sequencers are machines, and these work on principle developed by Frederick Sanger. So work on principle developed by Frederick Sanger. Okay, of course he also developed a method to determine the sequence of amino acids in a protein. Okay, so he also developed another principle that actually work in these automated DNA sequencers that will help in sequencing what are all the nitrogen bases or the base pairs on this fragment of DNA or on this, and so on, right? Yeah. So here the cloning resulted in amplification of this fragment of DNA. So we need to make many copies of this so as to use in these automated DNA sequencers. So by linking up these fragments, so once I took a long DNA, then breakdown, of course this will be done by using enzymes. Okay, so with some specific enzymes I'm going to cut this DNA into fragments, and each fragment, okay, will be linked to a vector, and this vector will be, okay, allowed to get into a host, and when the host multiplies, the vectors will also multiply along with that, and the DNA linked to vector will also get multiplied, so amplified. So I say cloning amplifies each piece of DNA. So that after amplification, we are going to use these automated DNA sequences to find out the sequence of base pairs on this fragment of DNA. So like that for all the fragments of DNA, right? So that's about the two methods which are followed for finding out the sequences of DNA. So once this is finished, so we have come to know a lot of information. So once we have finished the DNA sequencing of a human, so let's see, there are the salient features of human genome project. The salient features of human genome project. Now we came to know that the human genome, so it will be having, so human genome, so it has around 3164.7, okay, these many million base pairs are present. So 3164.7 million base pairs are present. And then later we came to know that, of course, during that human genome project, average gene, so the average gene, okay, consists of, or I'll say, on an average, a gene consists of 3000 bases. So it consists of 3000 bases. Okay, so averagely a gene will have 3000 bases, right? And then of course the size of the gene greatly varies, and we came to know that the largest right gene, largest human gene, so the largest human gene is dystrophin. So the largest human gene is dystrophin gene, that will be having, okay, that has 2.4 million base pairs. Okay, so it has 2.4 million right base pairs are present in that gene. So I'll say million bases, right? So dystrophin, the largest human gene, that has 2.4 million bases. That is what the information we have come to know. Then the total number of genes estimated. So total genes estimated. So these total genes that were estimated were, okay, around 30,000. 30,000, but earlier we thought that there will be around 80,000 to okay 1,40,000 genes are present, but to our surprise we came to know that that many um, genes are not present, that is from 80,000 to 1,40,000. So it had only 30,000, okay, genes, estimated around 30,000 genes. And then let the almost all, that is 99.9% of, okay, the nucleotide bases are same in all humans, so that all humans have, okay, 99.9, okay, percent of nucleotide uh, bases. Okay, so in all humans, so this is, it means that only right, the 0.1% makes difference between all of us. Okay, then the next one, the functions the functions of genes. Of course, 50% of whatever we have come to know. So the 50% of discovered genes, 50% of discovered genes, so their function is unknown. So their function is unknown, we don't know, okay, their function. So that is 50% of discovered genes, we do not know their function. Then we came to know that less than 2% of genome, so less than 2% of genome codes for protein. So it codes for proteins. Less than 2% of genome codes for proteins. Then we came to know there are, okay, a very large portion of a human genome has repeated sequences. Large portion of human genome has repeated sequences. So it has repeated sequences. So repeated sequences means, say suppose GCCT, again it is repeated GCCT again. So right, so it repeated many times. Later I'll come to that point when I discuss DNA fingerprinting. So how these repeated sequences and the mutations, okay, in them will actually help in identifying one individual from another individual. Okay, so large portion of human genome has right, repeated sequences, and these, of course, repeated sequences will be from 100 to 1000 times. So that repetition, okay, so the repetition, okay, will be from 100 to 1000 times. So the repetition can be, okay, 100 to 1000 times. And these repeated sequences do not have any direct coding functions. So they don't have any direct coding functions. So they don't have any direct coding functions. Okay, and of course, but it will help us to know the chromosome structure, dynamics, and evolution. So, but helps in understanding chromosome structure, helps in understanding the chromosome structure, the dynamics of chromosomes, and even evolution. So will okay, help. So those repeated sequences will not, I mean, they don't have any direct coding functions, but they help in understanding the chromosome structure, dynamics, and evolution, right? So that's some salient features. Now another one, the chromosome 1 has the most genes. Chromosome, of course, 1, this is the largest chromosome out of all the chromosomes in human beings. So this chromosome 1 has, okay, the largest number, or the largest number of genes. So that will be around 2968 genes are present. So chromosome 1 has 2968 genes. And coming to the chromosome Y. So this is the smallest chromosome. Chromosome 1 is the largest, chromosome Y is the smallest, and this one is having 231 genes are present on it. So we identified 2968 genes on chromosome 1 and 231 genes on chromosome, okay, Y. So the biggest and smallest chromosomes in case of human beings. And then, okay, scientists also identified about 1.4 million locations. Okay, so here around 1.4 million locations. So they have observed that there is a, single base, okay, DNA difference. So I say single base pair difference. So around 1.4 million locations they have found, okay, single base pair, okay, base pair difference, and that what we call it SNPs. So SNPs, that stands for single nucleotide polymorphism. Single nucleotide polymorphism. So let's say, right, there is a sequence like CGC, ATC, some sequence is seen. Now another sequence also has same, just you can see. So in this case, all the sequences are same, just one is different. So if you see another sequence, okay, so GCCTA something, you can see in this case, okay, at that point there is difference. So that's what we call it, single base pair difference we see. And so that is from person to person. When we see here, so in one person the same sequence, okay, but at one point there is one base difference. So that one base difference, that's what we call single nucleotide polymorphism. Okay, so single nucleotide polymorphism is seen. So right, these SNPs pronounced as SNPs, SNIPs, okay, and they of course occur in human beings, so they are called single nucleotide polymorphisms. Fine. So that's all about the salient features of Human Genome Project, and this information actually helps in finding out treatments for some diseases, okay, and even tracing human history also it can help, right? So that's about the Human Genome Project. So with this I will end today's session. In the next session we'll see application and the future challenges, and we'll also see about DNA fingerprinting.